Hi everybody, we're going to be doing Lab 11, which is Kingdom Animalia. We're going to be going over embryology and 3 phylums today. The phylums are Porifera, Cnidaria, and Ctenophora. They say animals evolved around 590 million years ago. All animals that we know of are multicellular, which means they're made of more than one cell, heterotrophic, which means that they make their own food, that they don't make their own food, that they get it from another source, and they're eukaryotic, which U meaning true, K meaning nucleus. It basically means that they have a nucleus and they have compartments. They're part of the supergroup Opisthocanta under the kingdom Animalia. In order for us to understand animals and their classification, we have to start at the very beginning. So in this lecture, we're going to talk about essentially how they develop. All animals start as what's known as a zygote or a single diploid cell. And then they're going to undergo what's known as mitosis or cell division. So when we look at animals, we can basically classify them by different characteristics. We have the 3 main basic characteristics that we classify them at. The first is, do they have or do they not have tissue? And an organism can be made of cells, but the cells do not have to form tissues. Tissue is a group of cells all working together for a common function. If they do not have tissue, it's parazoa. If they have tissue, it's eumetazoa. Another way to classify them is through body symmetry. Do they basically have mirror images of each other? If they do, it's bilateral symmetry. Can you cut planes made through a center that produces identical pieces? Them, goodbye lordy, they are radial symmetry. And some organisms have no symmetry at all. And the last is embryonic development. And we're going to talk about the different stages of embryonic development in exceeding detail. But basically, by the end of it, we will be looking at what parts of it produce what organs, and the whole, the blastopore, whether it becomes the mouth or the anus. Besides these basic characteristics, there's other ways to classify animals. If they have body cavities, basically we're going to look at animals and they either have what are known as germ layers, these layers of cells, and there's either 2 or 3 germ layers. If they have 3, they can develop what's called a coelium. This is a fluid-filled space, and it's between 2 of the germ layers called the ectoderm and endoderm. So we will look at whether it does not have a coelium, or if the coelium is put together in a way that makes it partial or complete. And the final is the type of digestive system. It can not have a digestive system, or it can have a digestive system. It can be a complete digestive system, which is called a tube gut, similar to what we have, where it has 2 openings, a mouth and an anus. Or it can be what's known as a gastrovascular cavity or a sac gut, where the digestive system only has 1 opening, and both food and waste get taken in or removed from that 1 opening. If you noticed in the PowerPoint, there are 2 asterisks, and those are the 2 that are not the 3 basic characteristics. They are characteristics 4 and 5, which are not the 3 basic characteristics. So basically what we're going to look at today are simple invertebrates. Invertebrate is something that doesn't have a backbone. And so when we look at animals, we know all animals are multicellular, and the very first division that we look at is, does it have tissue or doesn't it have tissue? Of course, if you look, Parazoa, there's no tissue, and you'll see our first phylum that we're going to look at today, the Porifera, or the pore-bearing. An example of this is a sponge. The Medusa is the next, and there are two examples of that. You can have bilateral symmetry or radial symmetry. And we are going to look at radial symmetry today for the most part. And so the Cnidaria, the jellyfish, the Hydra, the coral, and the other things that we will look at fit under there. And the last we'll look at is phylum Ctenophora, which is a comb jellyfish. I do realize that the handout said that we were going to talk about flatworms, but flatworms will be moved to the next time. So we are looking at the simplest of animals today, these three phylums, and you will continue to see this outline as we continue to move through the different animals. So our first characteristics is, does it or does it not have tissue? So this is looking at a bit of a simplistic phylogenetic tree, and it's showing the ancestral protists that we believe is a common ancestor with the choanoflagellates, and that at some point they diverged 707 million years ago to being without tissue and with tissue. So you have the Porifera over there, aka sponge. The Medusa, which you can see that they believe that they diverged about 680 million years ago, is both diverged into the Ctenophora and the Cnidaria, which are the two things that we're going to look at today. So, does it have tissue or does it not have tissue? That is one of the first questions you will ask yourself. So next we're going to look at the basic characteristics of embryogenesis. Embryogenesis. There's 5 steps here. The first is fertilization, and this is a process of a single sperm combining with a single egg to form a diploid zygote. From there, that zygote will multiply through mitosis or cell division, which is known as cleavage. During cleavage, you will get 2 processes. Compaction, which basically means as it sounds, cells are getting closer in contact with each other. And then a formation of a cavity, which is cavitation. Then we're going to move on to gastrulation, which is the arrangement of a cell that will create a blastula that essentially will help to create and differentiate the embryonic tissue layers, known as ectoderm, endoderm, and mesoderm. Then we'll get into segmentation, which is a division into repetitive segments. You might notice this in yourself. Our trachea, or even our spinal cord, or our vertebrae. Not the spinal cord, but the vertebrae, the bones. They're repetitive segments, or even in a regular worm, which we'll look at later. And then organogenesis, which is the process of organ and tissue formation. This happens via cell division and differentiation. And just to let you know, gastrulation and organogenesis together combine to morphogenesis, which is the biological process that results in an organism's shape and body organization. So this is what we will be talking about right now, and we're going to start with fertilization. So, if you look over here, we have an egg. It has different components to it. If you look at the zona pellucida, you'll notice that this is the area that helps guide the sperm to the egg. And this is separated by an area called the periphytallene space. And you can see within the space there are protein receptors. Now, on the inside, you'll see that there is a nice layer that's called the vitallene layer. Now, what happens here is that the sperm comes and finds the egg, and it does this by being drawn to it by the zona pellucida. The acrosome, which is on the head of the sperm, is going to come in contact with this area, and it is going to release hydrolytic enzymes. Now, if you remember from General Bio 1, hydrolysis is the process of using water or hydrogen to break bonds. So essentially, this is breaking down the area that it's coming in contact with so that it can essentially get into the cell and fertilize it. The acrosome will react with the perivitallene space. And then finally, the plasma membrane of the sperm and the egg will fuse. And then the sperm nucleus will enter the egg. Now, the cortical granules that you see here fuses with the egg plasma membrane, which renders the vitallene layer impenetrable to other sperm. So it prevents polyspermy, which is having more than one sperm fertilize an egg. So this is a process of fertilization that will create our diploid zygote. Now for cleavage, what we'll have is cleavage is basically another way to talk about mitosis. And so this is the first step, and you have the zygote, and then you'll go through, and I'm just gonna erase that stupid one stage thing. I don't know how that got there. And you'll see that it's basically multiplied several times to make an 8-stage clump of cells. AKA will make a multicellular embryo. And then over time, it will create a blastula, which is a multicellular stage that has like a ton of cells. And this 128 cells essentially will have the blastocyst cavity, which is that inside area that's actually filled with either fluid or yolk. So if we want to look at this, and we want to actually look at every step of this amazing process, we can start with ovulation, making the oocyte. That's the egg. And then it's going to come in contact with the sperm, and it will fertilize. And then essentially, after fertilization, you get the formation of a zygote, and you can see the sperm cell nucleus and the egg cell nucleus coming together, merging. And then you're going to start getting cleavage. So you get cleavage, which is mitosis, and you have 2 cells. That's the first group of cells. And then you get more cleavage, which is the 4-cell stage, and then the 8-cell stage. And then you get between 16 and 32, normally 32 cells. You get the morula, which is this clump of cells right before we create the blastula. And the blastula will essentially differentiate and have that area in the middle Here they're calling it a blastocyte cavity because this is comparable to what you would see in human. A little bit different than what we would speculate we would see in a simple old sponge. So, if you see here, you got some stem cells or embryoblast cells at the top. You have some troploblasts, which will actually be these outer layers. So this looks a little bit different than what we speculated earlier, but you know, it's a little bit more complex because it's looking at something more similar to a person. So any cells that result from cleavage is a blastomere, and this area, this fluid-filled blastocele that you see there, the function of it is basically to help permit cell migration during gastrulation and to help to prevent, it permits cell migration during gastrulation and prevents the cell beneath it from interacting prematurely with the cells at the top. So this does have polarity where the cells down here, you don't want them interacting with the cells up there. So this is kind of showing you what it looks like, and I just thought it was a super cool picture. I was looking at some journal articles and thought I would share it with you. It shows a cartoon image and a real image above it. And so you can see here that this is our original diploid zygote. One cell here, one cell here. 2 cells, 4 cells, then 8 cells, and you're starting to see that this has kind of a flower orientation. And we're going to look at this in more detail here in a second because you're going to see that the symmetry is based on how the cells divide and in what orientation. And this is where you'll start seeing compaction, where cells are getting closer together, you see, and they're starting here to make a cavination. So these plasmameres are kind of these non-differentiated cells, and the first cell fate decisions that they start making after that so you see the green that's around the outside is the trophectoderm, which essentially will be a membrane or the placenta. And then these inner mast cells here are going to differentiate into different germ layers. And so they're going to be basically what are later to know as pluriblasts or embryoblasts, which are structures of the function of the fetus. So you can see you have some purple here, and you're starting to see blue and red in the middle. So the blue part is going to be the endoderm layer, and this is going to be the primitive endoderm, or what will end up being the yolk sac. And the pluripotent or epiblast cells down here will end up being the embryo and essentially the ectoderm and mesoderm areas here. But this is a little bit more complex than the ones that we're looking at today because this is more comparable to what's in human. So when we're looking at gastrulation, that was just cleavage because we were forming the blastula. Now we're going to take the blastula and cause gastrulation. So review, zygote, 8-stage, cleavage. You have this hollow blastula, and based on where the cells are located and what's on each side, it's going to establish polarity in the body axis. So this is the blastocoele area that's in the middle. It's a cross section of it. And then you're going to see that you're going to start getting this kind of invagination that happens there. Things are actually pulling it and contracting it to get to the top. And this is going to start establishing germ layers. So you'll notice in here, it's a very simple one. You have the blastopore, which is this opening that happens. This is now called the gastrula. You have the outside, which is the ecto, think outside, and then endo, thinking inside, is this part that is will end up being the digestive tract. And you still have the opening here, which is the blastocele. So let's look at how this works. So you have on the blastula, you have two areas. You have the vegetal hemisphere and the animal. And you can see this bottom one here. It starts to flatten out. And once it starts to flatten out, you'll notice that you're going to start getting some cells that change shape and start moving inward here. And this inward movement is going to create this opening. Right now, this opening is a blastopore, but as it gets bigger, you'll notice that it's coming or being this like ancestral gut. And so this is an archenteron. And you'll notice that at both sides you have this red area here, which are the mesenchyme cells, which is going to be part of the mesoderm. So they're breaking free and starting to become this particular layer. And then you can see at the top here, you even have some of them there, and they're starting to create these thin extensions to attach to the ectoderm so that it can start contracting to help elongate that archenteron. So at the bottom here, because we do have, here we have three germ layers. We have the mesoderm, the ectoderm, and the endoderm on the inside. This part, this blastophore here, will end up becoming the anus. And then at the top, because you have this mesoderm here, this will end up becoming the mouth. So this is more complex. This is what's known as a triploblastic. If you notice, triploblastic gastrula has three germ layers. It has the endoderm, it has the exoderm, and it has the mesoderm. If you're looking at the diploblastic gastrula, you'll notice it only has the ecto and the endo. It does not have the meso. And these are examples of organisms that you can look at that are classified as each. Now, the organization of the two is pretty straightforward, and this, I think this does a really good job of kind of telling what things are. So if you have a diploblastic, it keeps doing this, it has radial symmetry. If it's triploblastic, it's bilateral. This, if it's diploblastic, remember it's 2. If it's triploblastic, it's 3. Their cells aren't as specialized as triploblastic. There's no special transport system in diploid, and there is in triploid. No central nervous system. They can have neuron networks. Here they're going to have the integumentary and a hardy nervous system. No mesoderm. Mesoderm. One cavity, and it's called the gastrovascular cavity. And this one, you have digestive system lining, you have glands for digestive system. It's more complex. Has a sac-like digestive system, has tube. This talks about the different types of triploblastic. You have the cilium there, or it's absent, sorry. Here you can either have the pseudo or the full. And then of course the groups here. So that's just kind of an overview of looking at the two. Now, when we look at this, this is just kind of doing a review of what we already talked about. So we have a zygote. And if it has no distinct cleavage, you have the blastula, it goes through gastrulation, and then you can see No real mesoderm created, no germ layers. What we're going to be talking about even more next time is what if it has spiral cleavage or radial cleavage? How is that going to affect, creating the mouth first or the anus first? And then from there, looking at their digestion. So we'll continue on into the triploblastic more next time, but most of what we're focusing on today is diploid. So during the process of gastrulation, we're going to look at making the digestive system, i.e., are we making the mouth first or are we making the mouth second? And this is a basic characteristic. So it's either a protosome or a deuterosome. And so if you look, the protosome, you'll have them where they are aligned basically over each other. So you can see that if you have something here, it's going to be right underneath it. Where this, they're off to the side. They're at an angle. So if they're right over each other, they're going to be a spiral cleavage, and it's called the determinant, and that it means they're going to make the mouth from the blastopore first. Think pro for first. If they're aligned at an angle, then essentially you're going to notice that the blastopore here is going to essentially make the anus, and the mouth will be made second, which is the deuterosome. Do mean if you think deuces as in twos when you're playing poker or whatever. So that means that the mouth is made second in this case, but the blastopore actually makes the anus. So in the protosomes, you're looking at mollusks, annelids, and arthropods, where in deuterosomes, you're looking at like echinoderms and chordates, more higher level. We're considered a deuterosome. So if you wanted to get a better idea of what that cleavage looked like, spiral, you can see it's over the side. So when you look down at it, the original is here and the new one is off to the side. Where radial is going straight up, so when you look at the first one, or the first one is here and then the second one is higher up. So that affects the polarity and the symmetry of the organism. Now, when you have those three, so the next thing we can do to classify organisms is look at, do they have a body cavity? So some animals that will have 3 germ layers, the triploblastic, will develop what's called a coelom. And this is a fluid-filled space, and it's between the ectoderm and endoderm. So the coelomate, which is the first one that we're going to look at here, which is a flatworm, you'll notice that you have the endoderm here, the exoderm here, and the mesoderm in the middle, and there is no fluid-filled space. So this means that it has no coelom. Now the pseudocoelomate, which is here, which is a roundworm, you'll notice that you have the ectoderm on the outside, the endoderm, and then the mesoderm here. It's not a true coelom. They do have a fluid-filled space, but the digestive tract is not covered with mesoderm. So if you're looking at here. So it's called a pseudocoelomate. A actual coelomate, which is here, and annelid, you'll see that the endoderm here and the ectoderm, and then you have the mesoderm, and you actually have that layer of mesoderm covering both the coelom all around. Here, it's only on one side. That's why it's pseudo. Now, in addition to that, you can look and go, okay, animals, do they have a gut? Do they not? They don't have a gut. Basically, what's happening is they're doing everything by intracellular digestion. They're either getting stuff to form food vacuoles, either phagocytosis, you go out and just grab a bunch of stuff and break it down in, or pinocytosis, that's how they pinch stuff off to get water in. You can research those more. We learn about those in General Bio 1. and they're broken down by lysosomes, but basically everything just kind of comes in pores and goes out a big opening. Now in a gut, it's going to come in a mouth and go out an anus, and there's either complete or incomplete or complete. Basically, the incomplete means it's just coming one way, in and out. Where complete means it goes in the mouth and actually comes out the anus. So it's like a tube, where this is like a sac. Now, the next process is segmentation, and this is just talking about being able to have or create segments. So in the body, it's pretty straightforward. So you can see segmentation and the worm here. Every single little thing here is repeated a segment. You can see the segments in the arthropod, including in the arms. And then even in the fish, the chordata there, you'll see it in muscles and you'll see it in the backbone. We have it as humans. It's in the trachea. It's in our backbone as well. So this is just kind of these repeated units over and over. And the last part is the organogenesis, which is making the organs. And you essentially make them, or they originate from different germlines. So the ectoderm over here, you'll see, forms the epidermal layer of the skin. Over here, it's the midline or the neural ectoderm will form the nervous system. You have the mesoderm here that will form bone, kidney, blood, gonads, and connective tissues. Gonads being reproductive cells. And endoderm here is the lining of the gut, the liver, and the lungs. So that kind of, and you still see the blastocyst that's in here. Vegetative pole here, animal pole up there. So this just kind of gives you an idea of looking at what germ layers and what systems are related. And you see that the mesoderm is responsible for the circulatory, skeletal, and muscles. The ectoderm is important for the integumentary system and the nervous system. And the endoderm is for the digestive tract. Now, this is another basic characteristic is body symmetry. And you can see in the porifera, the sponge, there's no symmetry at all. Where in the Cnidaria and the arthropod, you can see symmetry. And with the Cnidaria, you can see the radial because it has a center point and you can cut it into equal parts. Where the bilateral symmetry with the arthropod is the mirror image, which is what we have. Now, when we're looking at determining the body axis, how do we do that? And there's both extrinsic and intrinsic and yolk polarity factors. So cytoplasmic determinants are very cool. Basically what happens is that your egg prior to fertilization will have messenger RNA and proteins from your mom's genome. They're not distributed symmetrically in there. They are asymmetric. So one side has more than the other. So after you start getting cleavage division, then basically you're going to get two different resulting cells that differ in this intrinsic information that you have gotten from your mom's genome. So this is going to lead your cells to have different fates. For instance, they've done a lot of research on Drosophila, fruit flies, and there is a factor that they have looked at for the cytoplasmic determinant, which is called bicoid. And it's present in this concentration gradient across the unfertilized egg. And so the region that has the highest ends up becoming the head. The region with the lowest ends up becoming the tail of the embryo. And that's not much different than what happens with yolk polarity. So if you have an egg that has a large amount of yolk, well, if you have ever had an egg, you know the yolk is the heavy, dense part of the egg. So gravity is going to pull that to one side of the egg or the bottom of the egg. And so now you have asymmetrical distribution in the egg. So the region with very low yolk will become the head, and the region with high yolk will become the tail. And for more complex animals, because the yolk is things like amphibians and reptiles, birds and fishes, right? As you can see, I gave you an example above for Drosophila or fruit flies. Where we fit in is this induction or cell-cell communication, and it's one of the 5 essential developmental processes. And it occurs during development of all animals and plant species. And basically, it's a prime example of extrinsic information that's going to lead to different cell fates among individual identical cells. So basically, the embryo doesn't have any cytoplasmic determinants. They just have and they have very small amounts of yolk. So the induction process is a responsibility for establishing the body axis in this cell-cell communication process. Now let's look at the different phylums. So the very first one we're going to look at is Porifera. This is the oldest and simplest animal. And so we are essentially looking at sponges. So there are about 500 different species of sponges. They do not have true tissue. There's no symmetry. They contain groups of similar cells. They are filter feeders, which means they are filtering out or bringing in water and just taking through diffusion things out of that. Most of them don't move around. They're sessile. They're dioecious, which means they can be male or female, or monoecious. They can have male and female in both, depending on the type of sponge. In situations where they can be having a tough time, they can actually form gemmules to help with reproduction, and they can produce sexually or asexually through budding. So the different parts that we have here. So you have a spongocele here, which is this central cavity. The osculum, which is the opening at the top, that water gets pushed out. Now water comes in through these pores, this inhalant pores, and those pores are made up of cells called porocytes that are going to bring the water in. And the ostia is that area in the cell right here. The pentagoderm is the cell covering on the outside, and you can see the little dots, the little things that are coming are the spiracles. These are supportive structures. They're sharp. They're normally made of calcium or silica. The amoebocytes are over here. They're kind of pretty cool. They look like amoebas, and they're there to help store, digest, and transport food and waste or give rise to bud, but they're also there to help form the skeletal elements we know as spiracles. And the choanocytes are in here, and they help to trap food particles when things come into the spongocele. And this mesohyl is the skeleton formation. So when we look at these, if you're looking at something like that, the ostium is that opening at the top. You can just kind of look. Now, if we cut these all in half, the spongocele, you'll actually find it in there. I have it here because you can kind of see it there a little bit. And the spiracles are hard to see on here, but they're the little things that are here. And this is a picture just to kind of show you how it moves, because you can see that it goes through these pores, these porocytes that have the ostium that's in there. And then the spongocele is all here, and then the water ends up going out there through the osculum. And it talks about the different kinds of cells that you can find in there as well. Now, the exoskeleton is the non-living part of the sponge, and we talked about one form of it. It's just the spirals, and you can see that here. Sorry, this is spelled wrong. S-P-I-C, spiracles. The spicles. And so these spicules here, they're rough if you were to pick this up. where the spongins, this is really smooth. It's like the sponges you're used to cleaning with your house. So they're composed of different things, and it's different strengths. You can even look at these underneath a microscope. They look very pretty. And so these are the glass spicules or spicules. Now, we can also look at the Cnidaria, which are diploblastic, which means they have 2 layers. They have radial symmetry. They have a gastrovascular cavity. And these are interesting because they have 2 body forms. They have a mobile form called a Medusa, which the opening and tentacles face down. Perfect example of this is this guy here. And then they have the sessile form, which is the opening and tentacles face up. So you can see these are going up like that. Some members alternate between these 2 body types, depending on the stage of their life. Some of them don't have a life cycle, so they just stay in one. They can reproduce sexually or asexually by budding. They're cool because they're able to sting their prey. And that makes them essentially immobile. And they were the first animals to actually have a nerve net, which is the earliest form of a nerve. And so the different 4 groups that you can see, you have the Anthozoa, the Cubozoa, the Hydrozoa, and the Scyphozoa. So we're going to look at a few of those today. And this is just showing you the difference between the polyp, where everything goes up, and the Medusa, where everything goes down. So I have a few videos at the end, and the reason why I did it for these, I haven't done it for all of them because I really didn't feel like some of the things, for instance, if you're looking at a plant, to me, it's not much difference between watching a video of looking at a plant and looking at a picture. But for some of these, I really do feel like they're important to review. And so for you to see like even a dead specimen where they're able to move things around versus just a 2D picture. So let's start with the moon jelly here, the Scyphozoa, and then we're going to go through the Anthozoa.
So animals in this phylum are pretty interesting in the fact that they're among the most primitive true multicellular organisms. So in some of your labs you may have looked at single-celled animals, things like amoeba or paramecium. These ones are multicellular, and they're the first to display what's called tissue-level organization. So they have tissues, in other words, similar cells that are grouped or aggregated into specific patterns or layers, but at this point they still don't have true organs. Now, cnidarians have 2 distinct body forms. I'll be showing you actually a couple of different kinds. They have what's called a polyp form, which is usually sessile, or meaning that it doesn't swim around or move around. Or they have what's called a Medusa form, which is a free-swimming animal. And in this case, the jellyfish, in this stage of its development, is displaying the Medusa form. Now, some cnidarians display both types of forms during their life cycles, while others display only one. We're looking at a jellyfish. This is a species that's called the moon jelly. The species name is Obelia aurita. So the moon jelly is a really good example of what's called a true jellyfish, or animals in the class Scyphozoa. They're kind of neat animals. The name is appropriate. They're soft and squishy like jelly. They feel very jelly-like, and they're made up of anywhere from 94 to 99% water. And like other cnidarians, like all cnidarians, they display what's called radial symmetry. So to understand radial symmetry, we can think about a wheel. So if you think of a bicycle wheel, for example, you've got the round part on the outside, and then the wheel is divided by a series of spokes, okay? And in this case, the true jellyfish display what's called tetramerous radial symmetry. Tetra means 4, so it's basically a radial symmetry made up of multiples of 4. And we can see that right away in a couple of the structures, these sort of whitish horseshoe-shaped bits down on the bottom, and also these longer pieces right here. We can see there's 4 of those, so everything on this animal is made up in multiples of 4. Now, all Scyphozoans, all true jellyfish, have both of the two body shapes that I mentioned earlier, so the polyp and the Medusa form. In this case here, we're looking at a mature or adult animal, so this is the Medusa form, what we would think of as sort of a typical jellyfish. So we're mostly going to just focus on the external anatomy of this animal. This is the underside of the animal here. And the main part of the body is this, well, it looks round and flat here, but in life it would actually be curved like this, sort of parasol shaped, okay? And that part is called the bell. Okay, so most of this flat surface here is called the bell. And a hydrostatic skeleton, so a water-based skeleton, is basically what allows it to maintain its shape. So obviously it doesn't have bones, there's no hard shell like on an insect, so it's just a water skeleton that keeps its shape. So the outer rim of this animal, all on the outside where it's a little bit whiter, that's called the margin, okay? And the margin is divided into a number of sections, and we can sort of see those. We can see there's a point here and it sort of dips in and dips in and then in and then in and then in, and we can go all the way around the margin like that to see these different sections. And if we counted them, we would actually find that again they're there in multiples of 4. So again, the tetramerous radial symmetry. At each junction, so at each point between sections, we find a structure that's called a rhopalium. So a rhopalium consists of a few bits. You find a statocyst, which is used for balance. In other words, it helps the animal sort of orient itself in the water. And you also find something called an ocellus, which is used to perceive light. So it's sort of like a really primitive eye. So the other thing that we find along the margin are tentacles. And if you look really closely, you'll see these really thin, filamentous, fluffy little things all along the margin. Those are, in fact, the tentacles. And they shouldn't be confused with these larger things. Most people see these long things and assume that these are the tentacles, but those are in fact what's called the oral arms, or the arms. So the way these guys eat is the tentacles, these little filamenty things, are used to filter tiny, tiny prey, and then the oral arms, which are actually sort of trough-shaped, if you can sort of open them right here. So these trough-shaped oral arms collect or scoop this tiny filtered prey from along the margin, and it directs it in to the mouth, which is sort of right in the middle there. There's a hole there that goes to the mouth. The mouth opens up to a short little gullet, which leads to the stomach. From the stomach, we have 4 gastric pouches, which is basically where a lot of the digestion takes place. And the gastric pouches themselves are hard to see. Most things are hard to see in this animal because it's clear. But we know where the gastric pouches are located because they contain something else. These horseshoe-shaped pale white structures here are the gonads, the reproductive structures, and those are located within the gastric pouches. So near those pouches we find what's called gastric filaments, and we can't see those, but what's important to know is that the filaments are lined with special stinging structures called cnidocytes, and the cnidocytes are unique to the cnidarians. It's where they get the name from, and those are used to sting and so subdue anything that this jellyfish or any other cnidarian would want to eat. So it subdues the prey so that they can easily bring it to its mouth and then eat it. So now we're looking at an animal called a sea anemone. Anemones are in the class Anthozoa, and these are marine animals, so they live in salt water, and you'll mostly find them in intertidal zones. You'll find them pretty deep too. They'll go up to 75 meters deep in the water. Now, unlike the moon jelly that we just observed, these guys only live in a polyp form, so you'll never find them in a Medusa shape. They only live in this sessile, so non-swimming polyp form. So that's something that is a little bit different. So the body of the sea anemone is divided into 3 main regions. First, we have the oral disc, which is this upper surface right here that contains the tentacles and the mouth. Next, we have the column, which is the cylindrical shaped main body region in the middle right here that houses all of the guts, so to speak. And then lastly we have the basal disc, which is the underside, the base of it, and that's what it uses to attach itself to a substrate. So it actually secretes sort of a sticky mucus. It's like a sticky glandular secretion, and that's what it uses to glue itself, and also they can glide very, very slightly on whatever surface they happen to be on. Now, if you've ever seen a sea anemone in life, you know that they don't usually look quite this squished. The column here inside, and we'll see them in a moment, there's a whole bunch of really strong muscles in there, and in death those muscles have all been contracted, so the animal is basically completely compressed. In life it would be much more stretched out and nicer looking. So the tentacles, coming back to the oral disc here, the tentacles are all of these fluffy looking things up at the top here. And each of these tentacles is armed with those specialized structures that I mentioned on the jellyfish, the cnidocytes. So those sting the prey or subdue the prey, and then food is drawn over these tentacles into the mouth, which is this obvious opening up here at the top of the oral disc. So this is an anemone that's basically been cut in half, so we can see what's going on the inside. So just to orient you, so here's the mouth up here, okay? So the mouth opens into the pharynx, which is this sort of muscular structure right up here, and then the pharynx opens to the gastrovascular cavity, which is this little cavity at the bottom. So that's where the digestion takes place, and then waste would come back out through the mouth, right through there. Another thing that we can see are the gonads, so some of the reproductive structures. And if we look, here's a good example right here. So you can see what looks like it looks like a little stack of coins. If you imagine tiny little coins all stacked one on top of another in a pile, so like pennies in a pile, those are the gonads right there. So those are the things that produce the sperm or the eggs. Now, so the sex cells are released from those, and they go right out through the mouth too, because that's basically the only hole this animal has. So fertilization happens externally. It happens actually in the water, and then the fertilized eggs hatch into little tiny larvae that float down to the substrate, and they turn into individual polyps.
Great, so that was the two first types of organisms we're looking at in the Cnidaria phylum. Now let's look at the Hydra. And so you can see on the Hydra, these are solitary organisms. They like to be alone. You can see on the side here, which they've kind of blown up here, that you can see their neural network there, and this is a very simple nervous system. You can see the tentacles coming out at the top here and the mouth being in that area, and this does have radial symmetry because it has a centralized location here. And if you cut it, you'll get that kind of symmetry there. Now it has a gastrovascular cavity, so everything kind of goes in and goes out here. And you can see it does have budding for asexual. You can see an ovary here. And these guys can actually go out and stun their prey as well. So you can see right here is the trigger. And inside is this structure, almost like an organelle that's there. And that has a thread wrapped around it, and that's what's called the nematocyte. Sorry. And so the cnidocyte is this structure here, the stinging cell. So basically the nematocyte that's in here will be pushed out here. You can see it has a barb and this thread that goes out. So this essentially is what the Hydra looks like. Now coral is a little different. It doesn't like to be by itself. So it generally forms colonies, and it's strengthened by calcium carbonate, and there are other materials that take various massive plate-like brushy and leaf forms. They are generally in the polyp state. You look at where the openings are again, and they're sessile though. And you can see here all these different holes. Here are all these here will give you an idea of what kind of, if it's polyp or if it's Medusa. And this is a honeycomb structure here, and this is a mushroom structure. Now, Obelia is another type of coral. It has a worldwide distribution. There's a couple places on Earth that they are not located Arctic. Just think polar parts of the world. They have both a poly and a Medusa stage, and the Medusa stage is often found offshore in plankton. And so in this picture, you'll get a really good idea of kind of how they're put together. So here you have the gonozoid, which is the breeding bud. And within the breeding bud, you actually have the Medusa bud. And that will leave through this aperture. And this helps grow by feeding, and it gets released and can go places. You can see the tentacles here on these little guys. Here's a feeding bud, gastrozoid, and you can think gastrointestinal. That's where it comes from because it's part of the digestive system. And these are also called the breeding bud is also called a reproductive bud. So this shows you essentially what its life cycle is like. So you can see it's part of a colony. It has that reproductive bud. And coming out of that reproductive bud will be a Medusa. And that Medusa will come and release eggs and sperm. They will fertilize and become a zygote, which becomes a blastula. And that is the pinula that will swim until it gets to a place where it will implant and grow into a new colony of Obelia. So I will tell you there is a misspelling in here. It drives me crazy every time I've watched it. And unfortunately, this is the probably third, I believe this is the third time I've tried to record this lecture. So I'm sorry if I don't sound the most exciting. The first two times I was. By this time, I'm kind of done. So yeah, hopefully you don't have to watch it more than once. But yeah, every time I see this video, I'm just like, they are. So maybe you'll get it. But I wanted to watch. The reason why I went ahead and put it, even though it had the misspelling, is because I really wanted you to be able to see the different stages and what these look like, because I thought the pictures were fantastic. So let's go ahead and start. So hopefully that gave you some insight onto the different aspects or the different parts. Sorry. I don't know why it keeps... I'm having a lot of technical difficulties. Okay. Sorry. So that is the last of the phylum Cnidarians. So now we're going to get into, and we're only looking at one type of Ctenophora, which in this is the comb jelly. Ctenophoras are a small phylum. There's less than 100 species. They look very similar to jellyfish. They have a row of cilia that look like combs. That's why they are called or named comb jelly. They lack stinging cells, but they can secrete sticky substances that help to trap their prey. They do have radial symmetry, although the digestive system of the ctenophores may seem like a gastrovascular cavity. They actually have an opening dedicated for waste. Therefore, they present the first complete gut. And so let's watch this video over the ctenophores. And I have this video, and I've done this just for the last couple because I feel like it's really important for you to be able to see them a little bit better than just on some of the two-dimensional.
Comb jellies are ancient animals and voracious carnivores that belong to the phylum Ctenophora. This includes around 100 species. Their radially symmetric body contains 8 rows of comb-like plates of fused cilia that beat in order for them to move through the water. The comb rows diffract light to create a rainbow display as they swim. Some comb jellies additionally exhibit bioluminescence, or the emission of light by chemical reactions. Their body is over 95% water. When taken out of water, they literally look like a blob of jelly. About half of the comb jelly species feed by the use of a pair of tentacles that do not sting, but instead contain sticky cells called chloroblasts. Zooplankton will stick to their tentacles, and they will draw the prey into their mouth. They can extend their stomachs to hold prey half their size. Other comb jellies have no tentacles. Instead, their mouth contains hooks to bite off chunks of prey and secrete a poison to paralyze prey. They will even feed on other comb jellies larger than themselves. While the majority of ctenophores swim, some species remain on the bottom, creeping along the ocean floor or living on other animals. Most possess a gravity-sensitive structure called a statocyst that gives them a sense of up and down in the water. They have no brain, but contain a loose network of nerves called the nerve net. Ctenophores typically prefer warmer waters in temperate and tropical areas near the surface in both shallow and deep habitats. These jellies can be quite invasive, and their swarms can drastically change ocean ecosystems. Overfishing has caused an increase in populations of ctenophores because of less predation. All comb jellies are hermaphrodites. Eggs and sperm are cast into the water and fertilization takes place. Their larva grows rapidly as their entire lifespan is only around a few months. A full-size comb jelly can range from a few millimeters to over 4 feet.
In the lab itself. Is a zygote haploid or diploid? And we hopefully know that it is diploid. Sorry, I don't know. I'm having a lot of technical errors right now. What is the name of the hollow ball of cells during embryonic development? And hopefully you realize that that is the blastula. What is the stage when the ball of cells start to fold inward and create an archipteron? That is gastrulation. Name the 3 germ layers in a gastrula. So it's the ectoderm, the endoderm, and the mesoderm. How many germ layers do bilateral have? 3. Are animals eukaryotic or prokaryotic? Eukaryotic. Define heterotroph. And you can go, cannot produce food. Basically, they take nutrients from other sources of either organic carbon, mainly plants and animal matter. What are invertebrates? They're animals that lack a backbone. What is which supergroup does kingdom Animalia belong to? Opisthocanta. What are the 3 main characteristics of classifying animals? And so hopefully you realize it's either whether they have tissue, their symmetry, and their embryonic development. So that's our pre-lab. So now that we finished that, let's go to the lab. And some of the lab is a little confusing. So we're going to try to do the best we can to get through it. There's extra pictures. I don't really get what they were trying to do, but I'm doing my best to help you all out. So the very first one is table number 1, which is animal classification terminology. So you're going to use the chart provided on the table to label the body symmetry of animals below and specify how many germ layers each have. And there's not a table here. So if you look, they basically have a picture. If you see this, they have a picture of the jellyfish and they have a picture of a butterfly. This is one. So they're asking you what kind of symmetry does it have and how many germ layers. So you figure out what phylum they're in, and then from there you can figure out, okay, if they're in this phylum, they have this many germ layers and they have this kind of symmetry. And you can actually look at them to see basically the symmetry they have. And then you're going to do that in your lab practical also with this butterfly, symmetry and germ layer. And what you will do with the other ones is you should be able to practice to say, does this have symmetry? Hopefully you realize no, right? And how many germ layers does it have? Does this have symmetry? How many germ layers? This is a fossil. Does this have symmetry? Germ layers, symmetry, germ layers, symmetry, germ layers, symmetry, germ layers. Just so that you can practice, right? And then they give you some idea, because then they say embryonic development, define the terms below and label with pictures shown which term. So it asks you what is a protostome and the deuterostome. And then underneath it, it's going to ask you on the picture which one is a protostome and which one is the deuterostome. So you're going to have to look at where is that hole normally located, and where does the mouth get formed first, and then do it from orientation. Body cavities, use the picture given, draw the body cavity, label the 3 germ layers, and describe how each body cavity is different than the other. So this is the one where they're going to label, no, that's not it. This is just awful. It's not a very good. The lab itself is good, but the pictures just aren't. So on here, you can go back to, I put a picture in there that has all the different germ layers. not the different, right here, the body cavities. So basically you're going to take the body cavities and you are going to draw each one and then label them with the different germ layers. For embryonic development, we're finally at table 2. So you see there's a lot of pictures in there that you're not going to, and let me fix this. This is for labeling. This one is, put the wrong thing in there. Protosomes versus your next one it says embryonic development, and it is going to the table. So you can see the table has a bunch of different cells. So they're asking you, how is the zygote formed? And you'll go back and you'll hopefully realize that they're talking about the different stages of the embryogenesis that I went through. It's asking you to define the term blastomere, how are blastomere haploid or diploid. I'm giving 2 models, a morula and a blastula. How would you decide which one without seeing the blastocele? And then it's having you, so all of these are here, and then you can see this is a morula and this is a blastula without looking at, they're not showing you the other sides though, without actually looking at the outside, and you should be able to tell the huge difference between how big it is and how many cells. And then once you get done, they have this, which is the gastrula model, and they want you to be able to label the different parts of it, including the mesoderm, the endoderm, the ectoderm, the germ layer, the blastopore, and finally the archenteron, which should be pretty easy. Even though we haven't seen one that looked exactly like this, you should be able to take the pictures I've already given you. And they do have the labeling on there, but be able to do that pretty easy. And they're going to ask you, and here's some of the, there it is. They do show you the different pictures on here, that you can use this as well, or the one that's in the PowerPoint to help you with the different cavities. Anyway, they're going to have you do the different systems and what germ layers. And so you can find that in the PowerPoint as well. So now they're at table 3, looking at Phylum Porifera, the sponge. So they're asking you to take the sponge. I have the one on here. Something similar to this guy, and it's asking you to label the following parts. It's then asking you to go to the different parts, both this and this guy, and then say what kind of endoskeleton that they have, or exoskeleton. And then it's going to use this and is asking you to view and sketch these guys. And then it's asking you to go to the Venus flower basket, a bath sponge, and the colonial sponge, and say what kind of endoskeleton is in each, or if there's a combination. And they're giving you some hints on a couple of these to make it maybe easier. And then it has a picture of this fossil. And this fossil was in the Silurian period, about 1.5 million years ago. And this is a stereo microscope that was used to look at 6 of the tiny ray spicules or spicules. So they want you to look at the tiny stars embedded in the matrix in the fossil, observe them under the best you can here, and then label them. And they're going to ask you about the type of material that makes up the sponge endoskeleton, the type of body symmetry that the sponge has. Sponge belongs to a group called Protozoa. What are the distinct characteristics of this group, and what type of digestive system do sponges have? Cnidaria is the next table that we're going to look at. And this, you just have to go through. It's not in good order, so it's kind of tough. It's asking if the Cnidaria is diploblastic, and it's telling you to draw the polyp body. And so let me see if they have that here. I thought they had. They don't show it, so you can use one of mine for the polyp body too. So that is the going up, right? It's similar to what a Hydra is here. And then the Medusa body is kind of like if you were to look at a jellyfish. And you're going to say how they're different. And then you're going to look at the jellyfish and you're going to ask what kind of body form is it. It shows you a picture. And then it's going to ask you to label the different parts of this on your handout. And then what is the function of the nematocyst that's in there. And then it wants you to observe and draw the Hydra with the bud. So this is the Hydra with the bud, or you can even draw this one. They do have a sea anemone. They have this little jellyfish here. And we went over all the different parts of that. And then we did go over the different parts of this. We don't have a ton of questions on there, but I think the video did a really good job. It's looking at there. That is showing you the polyp and the Medusa. And this is showing you the different kinds of coral. So it's asking you to identify and label the common shapes. And they're showing you some of this here. And then you can fill it in. There's just 2 in the pictures, which are the bigger ones. And then the body form of the organism found in coral colonies. And then it's going to this type here. It's a clone. It's a Cnidarian that forms colonies. It's a feeding polyp, and so they have pictures of it really nice here. And they have one in your handout, and they're asking you to label it. And you can see some of that here. And it's what does a feeding polyp need tentacles? Why does a reproductive polyp need Medusa buds? And then what you're going to do is for the whole jellyfish, sea anemone, coral, Obelia, and the Hydra, there you might want to do this as you go through because they have a table. So it's asking you different questions. So I would look at that table every time you go there to at the end of this so that you can differentiate between all of those. And this is the table. It gives you some of the questions, and then you can fill in the others. And then table number 5 is the Ctenophora, which is the comb jelly. So it's showing you this little guy here. He's really tiny. See, that's why I wanted to show you the videos, because even though he's kind of cool, he's not as cool as what it's like when they are actually alive. So they're asking you what kind of characteristics. Hopefully you took some notes on the video because they did talk, and I did talk a little bit about this. Hopefully it's easy to see as well. And then you're done. And so from here, there are some post-lab questions at the end, and then some reflection. So good luck. I hope this helps. And again, just a reminder, I do the extra long introductions for you to help you get prepared for your lecture exam. So make sure that you are also watching these to help with that. Have a great night.